The article of choice for this presentation is called Comparative Analysis of Affinity-Based 5-hydroxymethylation Enrichment Technique. Direct chemical modification of cytosine bases found in the dinucleotide sequence CpG is a common method of epigenetic regulation in the mammalian genome. The most common form of this modification is the addition of a methyl group to the carbon 5 position in the pyrimidine ring by a family of DNA methyl transferase enzymes to form 5-methylcytosine. The most widespread of modified cytosine bases, corresponding to less than 1% of cytosine bases in mouse and human tissues, is that of 5-hydroxymethylcytosine. Disruption of the TAT proteins results in globally reduced 5-HMC levels, a phenomenon also seen during carcinogenesis. Also, breakdown of TAT1 in embryonic stem cells lead to an increase in 5-MC over transcriptional start sites, alongside loss of 5-HMC at specific promoters and within gene bodies of TAT1 target genes. Knockdown of TAT2 in hematopoietic progenitor cells was found to agitate normal gene expression pathways involved in differentiation, resulting in a block of myelate differentiation.
Mouse strains. This research paper was conducted on adult male mice. Their livers were used for DNA extraction. Their small pieces of liver contained DNA following their digestion with proteinase K, proceeding to phenol chloroform. During this experiment, the data sets were compared to hybrid mice, also known in this experiment as B6CH3HF1. JBP1 protein affinity. To produce DNA fragments, the genomic DNA was sonicated. An amount of 1 micrograms of DNA was then fragmented, taken through a process and glucose-lylated, incubated with glucose-modified enzyme for 1 hour at 37 degrees. The DNA was further incubated for 2 hours at room temperature with capture beads, which were magnetic and contained the JBP1 protein. The beads were washed in buffer before being immersed in the elution buffer provided. Purification DNA fragments. During this process, DNA was extracted from frozen -80 degrees before purification, and an average of 500 BP was fragmented. Microarray hybridization. Proceeding after purification, the samples that remained was prepared wholly for microarray analysis using a whole genome kit. This was then labeled and hybridized to a whole genome mouse with chromosomes 4 and 9 and all of chromosomes 5, 6, 7, and 8.
JBP1 protein in 5-HMC marked regions. JBP regions showed a similar trend to those of HME dip and HME seal. However, it was much weaker. This was caused by the fact that the amount of DNA recovered for JBP1 was lower than for the other two. Over 50 cycles. It took over 50 cycles to reach the threshold for JBP1. This was much longer than for the other two techniques, which took about 30 to 40 cycles. The researchers found that the amount of DNA present was extremely low. The test was repeated and gave similar results. Purification. JBP1-based purification of 5-HMC enhanced DNA showed levels of enhancement relative to input DNA compared to those of HME seal and HME dip. Microarray data for JBP1. JBP1 showed almost no correlation between the probes at all distances. The results received resembled those from random data. 5-HMC patterns. JBP1 showed lesser response. Only 2,561 brain peaks were recorded. That was much less compared to the peaks received from HME dip, with 130,560 peaks, and HME seal with 161,142 peaks. 3 techniques of 5-HMC purification. Researchers compared how 3 techniques work to purify 5-HMC in mouse tissues. The mouse tissues contained high, whole brain, or moderate liver levels of modification. The techniques used are antibody-based HME dip, chemical capture-based HME seal, and JBP1 affinity-based. The antibody and chemical capture-based techniques are known to be quite similar in genome-wide enrichments of the HMC modification, and they show high levels of tissue specificity and low levels of inter-individual variation, whilst GBP1 affinity-based techniques is significantly different to those above. JBP1 not worked. The researchers found no clear reason as to why the JBP1 affinity had not worked, but they believe there could be several reasons for that. The main reason being that during the qPCR, the JBP1 was working at low levels when it should have been working at higher levels. Also, the JBP1 protein may have been biased to certain parts of the genome, as the JBP1 libraries were enriched over the TSS, making the results different to those of HME-DIP and HME-SEAL.